Hello everyone, welcome to lecture 2 on animal kingdom. So in last lecture we discussed about different characteristics on the basis of which various phyla has been divided into the animal kingdom has been divided into the various phyla. So some of these characters were discussed in the last class, and in today's lecture we will discuss the remaining one, and we will then also discuss about the characteristics of each and every phyla in details. So basically we see that in the last lecture we see that what are the bases of classification of animal kingdom into different phyla. So animal kingdom were classified into different phyla on the basis of their level of organization, that whether they have cellular level of organization. Like on the basis of level of organization, they were divided as whether they have cellular level of organization, or whether they have tissue level of organization, whether they have organ system level of organization or organ level of organization, and whether they have organ system level of organization. So on the basis of that, different phylums were classified. Like we see that the phylum Porifera has cellular level of organization. It does not have a cluster of cells that can be called as tissues. So basically we see that different phyla were divided into the different levels of organization on the basis of which we classified different phyla. The next property that we discussed was body cavity. So some phylum have true body cavity, which were called as coelomates, while some phylum have false body cavity that we called as pseudocoelomates. So they were called as pseudocoelomates, right? So we call them pseudocoelomates. And then there were some phyla that we discussed and which do not have body cavity, which we call them as acoelomates. So on the basis of body cavity, different phylums were divided into the different different categories like pseudocoelomates, acoelomates, or coelomates. Similarly, on the basis of germinal layers, so what we discussed in the germinal layers there were 3 germinal layers, right? So one was ectoderm, that was the outer one, then the inner one, which was mesoderm, and the third one was the endoderm, right? So in some phyla, both ectoderm and endoderm were present, while in some phyla only mesoderm was present. So on the basis of the germinal layers also, we classified, or different phyla have different properties. In some phyla, all the 3 germinal layers are present, like ecto, meso, and endo, while in some only ecto and endo are present. Then we will see how the different phyla are classified on the basis of notochord. We will see how different phyla are classified on the basis of the circulatory system. And today we will see how different phyla are classified on the basis of the metamerism or segmentation. Also, in the last class, we discussed on the basis of symmetry. And we discussed this is very important in the case that most of the questions are asked from the symmetry, because we discussed 3 different types of classifications on the basis of symmetry. The first one was the one which were asymmetric, that is, which do not have any kind of particular symmetry, like most of the sponges. So most of the sponges or the poriferans do not have any kind of symmetry. Then we discussed about the radial symmetry, right? And also we discussed about the bilateral symmetry. So the bilateral symmetry. So this was discussed in the last class, and from today, in today's lecture, we will focus on metamerism, we will focus on circulatory system, and we will also focus on the notochord, right? And after that, we will start for the different phyla, that what are the characteristics of each and every phyla. And there also we will relate to all of these bases of classification again. So basically, metamerism or segmentation is property of some of the organisms of some phyla. And in some animals, the body is divided externally as well as internally. So externally and internally, the body is divided into the different segments. And these segments are serially replicated, at least for some of the organs. For example, earthworm we all might have seen. So in earthworms, we have seen that the body shows the pattern called metameric segmentation. So this metameric segmentation is basically the serial repetition. So we can see that in the earthworm, you can see that these are the different segments that are present, which I am highlighting right now. So these are the different segments that can be numbered also, like we can number these segments. So there are different segments that are being present, and these segments are, and this phenomena is basically known as metamerism. So these separations are the septa, and this septa provide regional control over hydrostatic skeleton. So this is the property of earthworm basically, but in general, metamerism is basically the segmentation, and the segmentation of the earthworm basically is referred here to as metamerism. So in some of the animals, the body is divided externally as well as internally into segments, and therefore they are said to have proper segmentation, while in others the segmentation is absent. Then the next structure is the notochord. So as we see that we have three germinal layers, right? So we discussed about three germinal layers in the last lecture. The one is ecto, as I mentioned again. The second one is meso, the central one, and the third one is endo, right? So most of the internal organs are derived from this, external from this, and this mesoderm basically helps in creating the body cavity, true body cavity. So notochord is the structure. So this notochord is basically the structure which is derived mesodermally. So it has a mesodermal origin. So it has been derived from the central layer, that is, it has a mesodermal origin. It is a rod-like structure which is formed on the dorsal side. So this is really important. Firstly, it is derived from the mesoderm. So something that we need to remember here is that it has a mesodermal origin. Also, it is present on the dorsal side. So this is important, that it is present on the dorsal side, and it is a mesodermally derived rod-like structures. Then animals with notochord are called chordates, and those animals which do not have this structure are called as non-chordates. So as the name can be clearly seen, those which have this notochord are called as chordates. Those which do not have this notochord are called as non-chordates. So the phylum, the animals that belong to the phylum Porifera, echinoderms, that is Porifera, Coelenterata, Ctenophore, Platyhelminthes, SK helminthes, Annelids, echinoderms, they all belong to non-chordates, that is, they do not have this notochord, or this mesodermally derived rod-like structure is absent in these organisms, while in vertebrates that we will discuss later, like in aves, mammals, we have this notochord. So therefore, we belong to the category chordates, while the organisms that lack this notochord belong to the category, belong to non-chordates. For example, the animals belonging to phyla Porifera to echinoderms. The next basis of classification is the circulatory system. So we have two types of circulatory system. The one is open circulatory system. The second one is closed circulatory system. So we can see clearly that in open circulatory system, the organs are openly bathed with the hemolymph or the fluid. While in closed circulatory system, we have the system of small branches, that is vessels, that run into every organ and supply blood to the organs. So in this, the blood is not present in the body but circulate inside the blood vessels. While in this, the blood is directly bathing the organs which are present inside the body. So we have open circulatory system where the blood is flowing openly, while in closed circulatory system, the blood flows inside the blood vessels. So open circulatory systems are common to most of the arthropods and mollusks. So in arthropods and mollusks, we have open circulatory system, while if I talk about closed circulatory system, it is present in vertebrates, including humans. So humans also comes under the category of vertebrates, that comes under the category of chordates. So basically we are chordates, that is we have notochord, and then we comes under the category of vertebrate. We will see why we are called as vertebrates. And so humans comes under the category of vertebrates, and we have a closed circulatory system where the blood flows inside the blood vessels and then run into the organ. So this is kind of summary for whatever we have understood in the last lecture and in today's lecture till now. So we have kingdom. So kingdom is the Animalia. So this Animalia kingdom includes the animals that are multicellular, which are heterotrophic in nature, right? So on the basis of level of organization, we have cellular level, and then we have tissue, organ or organ system level of organization. So from the after level of organization, we can classify the organism on the basis of symmetry. So the organisms that have cellular level of organization are mostly asymmetrical. Also, they do not have body cavity, so they are acoelomates, and the phylum is the Porifera. So this is the first phylum that we are going to study today in today's lecture, where the animals that are what are the basic characteristics of Poriferans? They are multicellular. They have cellular level of organization. They are asymmetrical. Most of them are asymmetrical. They are acoelomates. They do not have body cavity present, right? So these are the basic characteristics of the phylum Porifera, and we have tissue level of organization, organ or organ system level of organization. So those organisms can then be classified on the basis of symmetry. So they may either have radial symmetry or they may have bilateral symmetry. So we understand that in radial symmetry, the organism can be divided into two equal halves from any axis that passes through the center. While in bilateral, there is only one axis that can divide the organism into two halves, right? Bilateral symmetry. So the organisms that have the radial symmetry are all acoelomates, and they belong to the phylum Coelentrates, that is Cnidaria, which is also known as Cnidarians, or Ctenophora, right? While the organisms that have the bilateral form of symmetry, they can be acoelomate, that is without the body cavity. They can have false coelom, so they have pseudocoelom. They can be called as pseudocoelomates, or they may have the true coelom, so they may be called as coelomates, right? So the ones which are multicellular, which have tissue, organ or organ system level of organization, which are bilateral, which are acoelomates, belong to the Platyhelminthes. Similarly, which are pseudocoelomates, belong to Eschhelminthes, and the organisms that are multicellular, heterotrophic, have tissue, organ system level of organization, have bilateral symmetry. They have true coelom as well, that is, they are coelomates, belong to phylum Annelids, arthropods, mollusks, echinodermata, hemichordata, and chordata. So we will learn about each of these phylum one by one. But here, the point that we should notice is that we have a star marked here as echinoderms. So why they are star marked? Because in the last lecture also, when I was teaching you about the symmetry, we found that echinoderms contains the organisms that has both radial and bilateral symmetry. This radial or bilateral symmetry depends upon the stage. So we can say that the adults have different symmetry while the larval stage has different symmetry. So depending on the stage of the organism, whether it is larval or whether it is the adult stage, they may exhibit radial or bilateral symmetry respectively. So this is a broad classification of kingdom Animalia based on the common fundamental characteristics, and if possible and if you are able to remember this table, this classification, this will help you a lot in understanding most of the characteristics of the phylum, of a particular phylum, right? So for today, we will start with the phylum Porifera, and so we will learn about the characteristics of phylum Porifera. So animals in phylum Porifera are informally known as sponges. So you can call the organisms that belong to phylum Porifera as sponges. So what basically Porifera means in Latin? The porous, Porifera means the porous, pore, and fere means to bear. So Porifera are the organism that bears the pore. That is, they have the pores, right? So these pores play a very important role in the overall functioning of the poriferans as well, that we will see how. So among the simplest of animals, so this phylum consists of the simplest animals, so they have the simplest animal group. Sponges are sedentary. Sedentary means they cannot move, so they are not mobile, they are actually sessile or sedentary, and most species are marine. So mostly they live in marine water, ranging in size from a few millimeters to a few meters. So these are the basic characteristics, that they are the simplest form of animals, they are sedentary, most of them are marine, their size ranges from few millimeters to few meters. Also, they have various body forms and shapes. As we see that the discussion, we see that they are mostly irregular in shape and therefore they are mostly asymmetrical. That is, they cannot be divided into two equal halves. So this thing came again. So as we will learn about each and every phylum, whatever we have learned till now will keep on repeating. So we need to remember that the organisms that belong to this phylum are mostly asymmetrical. However, there are certain exceptions to this. For example, Cycon or Leucosolenia are the examples of poriferans that have the radial symmetry. That is, their body can be divided through any axis into two equal halves that passes through the center of the body. So any axis that passes through the center of the body can divide the body into two equal halves. So this radial symmetry is present in the Cycon and Leucosolenia. And these are exceptions to the asymmetrical nature of the organisms of phylum Porifera. So you need to remember the exceptions as well in every case. So these you need to remember that some of the organisms like Cycon and Leucosolenia are radially symmetric. Then the very important thing is that they are filter feeders. That is, they filter out food particles that is suspended in the water, and they drew it through their body. So they take the food particle in the form of and basically they filter out those food particles which are suspended in the water as the water passes through their body. So therefore they are called as filter feeders, right? I have highlighted this also. So they are filter feeders, and as we mentioned in the previous discussion also, as we discussed, that these organisms lack tissues. Right? So these organisms lack tissues, that is, they have cellular level of organization. These do not have any well-defined group of cells that perform a specific function. So there is no group of similar cells. What is the definition of tissues? Tissues are basically group of similar cells that act as a functional unit, right? So they lack tissues and they have what? Cellular level of organization. That is where the cells, different cells play different functions, have different roles, and there are no group of similar cells that act as a functional unit together, thereby lacking the tissues system of organization. However, the sponge body, as I mentioned, that it contains several different cell types, and each of these different cell types have their own function which they perform in order to carry out the activities that is needed by the poriferans. So many pores perforate the sac-like body of a sponge. As I mentioned, the name itself, porifera, suggests that they are pore-bearing. So therefore, many pores perforate the sac-like body of the sponge. And very important that they have a canal system of varying complexity. So they have a canal system that allows the water to move through the body. So basically, this canal system allows the movement of water inside the body. This we will see how, right? So the interior of the canal is lined with the flagellated cells that also help in filtering out. And in a sponge, these cells are called as collar cells or choanocytes. So the flagellated cells that are present on the inner lining of the interior of the, so basically suppose this is a sponge. So on the inner lining, we have flagellated cells, right? Like this. So on the inner lining, we have flagellated cells. These flagellated cells are called as collar cells. So collar cells are the flagellated cells, that is, they have flagella, and this flagella also helps, like the beating of this flagella helps in the producing the water currents that flow through the pores. So these pores which I mentioned, which are present on the body of the sponge, are called as so these pores which are present on the body of the sponge are called as ostia, and these ostia are basically the openings, and these ostia open into the central cavity, and then the water moves out through the osculum, which is the upper opening of the body. Skeleton is internal and consists of tiny calcareous spicules, or they may be siliceous spicules, so they may be made up of calcium or they may be made up of silica, or fine spongin fiber located in the mesenchyme. So the internal layer, they do not have any mesoderm, but they have mesenchyme in which this calcareous or siliceous spicules are present that act as the skeleton. So these spicules provide the support to the body and therefore act as the skeleton. So the skeleton is internal in these organisms, and it consists of calcareous or siliceous spicules or fine spongin fibers which are located in the mesenchyme. Scleroblast secretes spicules. So these spicules are secreted by the cells which are scleroblast. So these are the cells that secretes the spicules. So basically, you can learn this also, that as I mentioned, that in these animals, basically different cells perform the different functions. So scleroblasts are the cells that secretes the spicules, and spongioblast secretes the spongin fibers. Right? Respiration and excretion takes place by the diffusion of gases. There is no organ system. There is no well-defined respiration or excretory system. Therefore, it takes place by the process of diffusion of gases through the surface of the body. So simply by the through the surface of the body, diffusion by the process of diffusion, respiration and excretion takes place, and excretory material is ammonia. Sorry for the mistake here, it's ammonia. Right? So the excretory material is ammonia. So ammonia is excreted out of the body by the process of diffusion. So the respiration and excretion takes place through the body surface. There is no well-defined organ for respiration or excretion, and the excretory material is ammonia. So this is the anatomy of sponge. As I mentioned that we have outer layer, then we have pores which are present in the outer layer, and then we have choanocytes or the collar cells which are present on the inner layer that lines the inner layer, and the internal cavity is known as the osculum, through which the water flows out. So we will learn about each of these one by one. So the outer layer of this sponge consists of tightly packed epidermal cells, and its wall has two layers of cells separated by a gelatinous matrix. So basically, they do not have any. They do not have any mesodermal layer, but the outer and inner layer of cells is being separated by the gelatinous matrix. So they have a gelatinous matrix that separates the outer and inner layer instead of the mesoderm, which is present in higher level of organisms. So instead of that, they have a gelatinous matrix, right, that divides the outer and inner layer. And then we have pores. So water enters the sponge through the pores. So we called these pores as, we discussed about this. So we called these pores as ostia, right? So water enters through them. Water enters through these pores and gets into the body cavity, which is known as the osculum. So water enters through the ostia, and the central cavity is known as the spongocoel. So the central cavity is known as spongocoel, and the water enters into the spongocoel here. So this is called as spongocoel. That is the cavity of the sponge, and it moves out through the osculum, right? So this is osculum. It moves out of the body through the osculum, right? So water enters through the ostia, then it comes into the spongiocele and it moves out through the osculum. Then we have choanocytes. So in the central cavity, the cells, sponge has a central cavity which is lined with these cells called the choanocytes that have flagella. So these choanocytes have flagella, and the beating up of this flagella creates a current that draws the water in through the pores. So basically we need to develop that water current through which the water enters through the pores, right? So the beating of this flagella helps in creating the water current that draws the water in through the pores and out through the body cavity, right? So this beating of flagella allows that water current to drawn and allows the entry of water into the spongiocele, and then it allows the water to move out of the body through the osculum. So this is the large structure of the choanocytes. So in the choanocytes or the collar cells, they have the collar like structure which has a flagellum, right? So this flagellum beats and allows the water to move inside the body. Also, these cells help in the phagocytosis of the food particles, which are being present along with the water. So these choanocytes basically phagocytize the food particles which are present, and they have an amoebocyte-like structure that allows the excretion also, right? So the movement of choanocyte flagellum also draws water through its collar of finger-like projections. Food particles are then trapped in the mucus that coats these projections, engulfed by the phagocytosis, and they are either digested or they are transferred to the amoebocytes. So basically you can clearly see here, the food is either digested and is removed outside the body, or it is being transferred to the amoebocytes where it is stored. So basically these amoebocytes are the cells. These cells, they can transport the nutrients to other cells of the sponge body. So amoebocytes are the cells that can transport the nutrients to the other body. Then we have structures that we discussed as spicules. So these spicules, we can see that they are made up of calcium or silica. So these calcareous or siliceous spicules or spongin fibers act as the internal skeleton. So you can see that the presence of these spicules are helping in providing the structure to these cells, and therefore they are acting as the skeleton, right? So they act as the skeleton for the sponge. So these are the different structures that are present. So we have the outer layer that consists of pores, which are called as ostia. Then we have choanocytes that line the inner cavity, and these choanocytes have the collar-like structure that helps in producing the water current. They have flagella for the beating, and they also perform phagocytosis of the food particles. If the food is not digested, they transfer it to the structure which is known as amoebocytes. These amoebocytes help in transporting the nutrients to the other cells of the sponge body. So this is how different cells perform different functions in sponges. So they have cellular level of organization, but we can clearly see that they do not have any digestive system or excretory system or respiratory system per se. So they lack tissue level of organization, but they have a cellular level of organization. So this is the structure of how the water comes into this. So basically water enters through we can see that the water enters through the ostia. So we have water here outside the cell, outside the body. It enters into the body through ostia, which are the pores. So water enters through the pores, and then it comes into the central cavity, which is the spongiocele, right? Through which it comes out to the osculum. Osculum is this outer pore. So this is the osculum. So water comes out through the osculum, and the water is then out of the body, right? So this is how the water is being taken up by the sponges. So they have water canal system. This water canal system is really important to understand because this is particular to this poriferans. So in water canal system, basically what is happening? Water is getting inside the body of the poriferans through the pores, which are called as ostia, which are the openings. It enters through the central cavity into the central cavity through ostia, which is known the central cavity is known as spongiocele. Then from spongiocele, it reaches to the osculum and the water is drawn out of the body. So this is happening in the water canal system of the polyphenols. Now, the very important thing is how the reproduction is happening in the sponges. So till now, what we understand about the sponges are that they have cellular level of organization, right? They are asymmetrical except some exceptions like cycle. They are asymmetrical, right? They are mostly sedentary. They are not much mobile. They are mostly marine. So they are mostly marine. Also, they have a very particular type of system, which is the water canal system that allows the movement of water. And during this movement of water, they also takes up the nutrients which are being present in the form of food particles in the different which is present by the process of phagocytosis. So basically, we learn about different types of cells also. So we learn about choanocytes, we learn about the amoebocytes, we learn about archaeocytes. So the archaeocytes we will learn now, that archaeocytes are being present in these. So sponges, now we will talk about the reproduction in sponges. So sponges are among the group of organisms that possess both egg and sperm. So they have both egg and sperm, and therefore they can be called as hermaphrodites, since both of the female and the male organs, like both male and female organs, are being present in the same organism. So they can be called as hermaphrodites. And they can reproduce either sexually or asexually. So they have both kinds of reproduction. So sponges can undergo both type of reproduction. That is, they can reproduce either sexually or they can reproduce asexually. Asexual reproduction in sponges is mainly by the process of budding, fission, formation of reduction bodies, and the formation of gametes. So we will learn about each of these one by one, that how the budding, fission, formation of reduction bodies or the formation of gametes help in the reproduction of sponges. Firstly, by the process of budding. So we know that what are buds basically. In this, the structure which is known as the buds is being formed. So buds are nothing but the protrusions, right? So these are the protrusions that protrude out of the normal body of the poriferans. So during budding, a small protuberance appears, right? So for example, this you can see that a small protuberance has appeared here. So outgrowth has arisen at the base. So this is the protuberance that appears during the process of budding. Then this outgrowth basically increases in size. So we can say that the bud grows in size. So this bud grows, gradually increases in size until it forms a complete organism. So when a complete organism is being formed, bud may remain either attached to the parent or it may break out from the parent and get attached to the new substratum. Since we call them as sedentary, that is, they are always attached to some substratum. So in that case, so a substratum must be present on which these are attached because they are sedentary in nature. So this is the substratum. So the bud may either remain attached to the parent body or it may get detached, right? So it gets detaches. So sometimes it detached and forms a new colony. So the bud may break out from the parent and it may get attached to the new substratum. So during budding, basically what is happening? Firstly, a protuberance is appearing. So a protuberance appears, which actually grows in size gradually and leading to the formation of complete organism. And the bud that is produced, so the budding produces colonies of sponges that can become quite large. So basically, this budding will lead to the formation of colonies, because what happened is, if this bud remains attached, then other budding may also happen, right? So in that way, there are many organisms that remain attached to the same substratum, leading to the formation of colony. So they may lead to the formation of colony, right? where different organisms are being attached to the same substratum, right? So different organisms attached to the same substratum or the base, right? So they may lead to the formation of colony or the colony-like structures. Budding produces colonies of sponges that can become quite large if the bud does not break out from the parent cell. The next is the process of fission. So we learn that one of the asexual method of reproduction is budding. The next is the fission. So in fission, basically what happens is that the parts of the sponge body are thrown off from the sponge body, right? So some parts of the body, like you can see in this figure also, a line of weakness is being created, right? This line of weakness leads to the splitting of some parts of the parental body and which are thrown out. So the parts are splitted and thrown off the parental body. So you can see that this line of weakness leads to the pinching off, you can say. It's simply the pinching off, you can say, that the parts are being splitted and they are thrown off the parental body, and these parts then develop into the new individuals. So this is the process of fission. Fission is basically what? Fission involves the dividing of anything into different parts. So this fission basically leads to the suppose this pinching off takes place due to the line of weakness. So these different organisms are being so this part is being created. This developed into the completely new organism. So this mode of reproduction is the fission. So this mode of reproduction is known as the fission, where the parts of the sponge body are thrown from the sponge body. The sponge is hypertrophied over a limited area, developing the line of weakness. So basically, it develops a line of weakness, and along this weak line, splitting happens. So, and this part is thrown off from the parental body. The part of the parental sponge thus thrown off develops into an adult individual, into a separate adult individual, and breaks off an osculum at its free distal end and gets attached to a suitable substratum. So you can see that these parts developed into the complete organism that has osculum. So this is the osculum, which is the osculum at the distal end, free distal end, and at the base it gets attached to the substratum. This new individual develops a new colony by the budding. So again, this new individual may lead to the formation of protrusion. These protrusion may develop into the new organism. So these individuals may again form such colonies by the process of budding, right? So budding is basically helping in the formation of colonies, while fission is leading to the reproduction by the formation of the line of weakness, where some part of the parental body is thrown out, and this splitted part is then developed into a complete new organism that even develops an osculum or breaks off an osculum at its free distal end. So this is its free distal end, right? At its free end, it develops the osculum, and on the basal side, it gets attached to the substratum. So it gets attached to the substratum. It finds a substratum where it can get attached, and at the free end, it has osculum developed, so it can take up the water and can throw out the water out of the body, the water canal system, and in this way it can survive, right? So this new individual develops. If this new individual wants to develop a colony, it can develop through the process of budding. So this new individual may develop a protuberance. This protuberance may grow in the size and may lead to the formation of bud, right? The next is the formation of reduction bodies. So it is a very unusual method, basically, which is found in the sponges. And some freshwater and marine sponges gets disintegrate during the adverse condition. So formation of reduction body happens usually during the adverse condition. So this is the mode of reproduction that mostly takes place during the adverse condition, where the sponges get disintegrate. So its cells get disintegrate because it only has cellular level of organization. So its cells gets disintegrate during the adverse conditions, and during unfavorable condition, the sponges collapse, leaving small rounded balls which are called as the reduction bodies. So these are the small cells. So panacoderm is present in the outer layer. Amoebocytes are the cells that helps in providing the nutrition. PYQs help in providing the skeleton. But during the unfavorable condition, what happens is that these collapses, all the cellular system even collapses, leading to the formation of reduction bodies, and spicules are, you can see that now not much further skeletals, they do not act much as a skeletal system. So they leave these small rounded balls, which are called as the reduction bodies. So the formation of reduction bodies takes place, but this happens during the unfavorable or the adverse conditions. The next is the formation of gamules. So gamules are the internal buds formed within the sponge body. So these are the internal bud, and it is a characteristic of both freshwater and some marine forms. So in all the freshwater sponges, this gamule formation happens, while in some marine forms also gamule formation may take place. Gamules eventually gets detached when the parent sponge is decayed. So these are the internal buds. So there is a difference between the budding and the gamule formation. So these are the internal buds which are formed basically inside the body, sponge body, which is formed within the sponge body. And eventually when the parental sponge gets decayed, these are exposed and gamules eventually gets detached from the parental body. And it helps the sponge tide over unfavorable conditions. It can withstand freezing and considerably great degree of desiccation. So basically, it has the power to bear the unfavorable condition as compared to the adult sponges. These structures have the power to withstand the adverse conditions like freezing or greater degree of desiccation, that is where the water is not available, as compared to the adult sponges. So adult sponges do not have that much capacity to bear that, but the gamules can withstand freezing and considerably they can bear a greater degree of desiccation also than the adult sponges. So basically gamules and as well as reduction bodies are the structures that are being formed during the adverse condition. So what is the structure of gamules? So gamules contain an outer membrane, right? They have archaeocytes cells which are being present. They have inner membrane. So this is the outer membrane. This is the inner membrane. They have archaeocytes and they have monaxon spicules. And the opening or the cavity is known as the micropyle. So instead of osculum, they have micropyle, right? So gamules are the internal buds which are formed within the sponge body, and it is mostly formed in the freshwater sponges instead of the ones which are present in marine, although they are formed in some marine forms also, and they are mostly formed during the adverse conditions. So till now we see different kinds of cells also, like amoebocytes, which are archaeocytes. We see choanocytes. These are the things that we need to remember. So we see different modes of asexual reproduction in sponges also, like what are the different modes? Budding and fission, which takes place during the favorable condition, where the protrusion happens. So protrusion happens, which grows in size, leading to the formation of new organism. In fission, what happens is there is a formation of line of weakness, right? So there is a formation of line of weakness here, leading to the splitting of the structures. These structures developed into the new complete sponge that is attached to the substratum. And then by the process of budding, it may lead to the formation of colonies. Now, formation of reduction bodies and formation of gamules mostly takes place during the adverse condition. So these are mostly the mode of reproduction, or we can say that these are the mode of survival also during the time of adverse conditions. So this was about the asexual reproduction that happens in the sponges. Now we will learn about the sexual reproduction in sponges. So we know that though some unisexual sponges species are also known, most sponges are monoecious or bisexual. What do I mean by that? What do we mean by monoecious? So monoecious, so it is a very confusing term because in this both the sexes, that is both male and female, are present in the same organism. So you may feel it little bit confusing if I compare it with the term dioecious. So dioecious means those that require two organisms as male and female individually, while monoecious are the ones that contains both male and female within the same organism. When the male and female organs are being organs. So when both male and female organs are being present within the same organism, they are called as monoecious. While if it is present in the two different organisms, then it is called as dioecious. So basically, you can call monoecious as equivalent to bisexual, that has both male and female organs within the same organism, right? Fine. So this is very important to remember that although there are some unisexual sponges species which are known, but most of them are monoecious, that is, they are bisexual. So if you will have the clarity about the terms, it will be easy for you to understand these terms and you will be able to relate it better with all the theory also. So it is very important to remember that monoecious, this might seem confusing also to some individuals, but monoecious are actually the ones in which both the organs, male and female reproductive organs, are present within the same individual, right? So monoecious, or they may be called as bisexual also. So we can say that although sponges are bisexual, bisexuals may be referred to as hermaphrodite also. Cross-fertilization occurs as a rule as the production of timing of sperm and ova are different. So we may call that why cross-fertilization is happening in this case. So what do I mean by cross-fertilization? We will also need to know that. So basically, when we are saying that both male and female organs are present in the same individual, what we will expect is that the male cells or the male reproductive organ will release the sperm, while the female will release the ova. Both of will undergo the fertilization. The fertilization takes place within the individual by the male and female sperm and ova which are being formed. But in this case, what happens is that process of cross-fertilization is taking place. So why cross-fertilization? Cross-fertilization means sperm which is present in one organism is fertilizing the ova of the other organism, irrespective of the ova that may be formed within the same individual also. Similarly, the ova of this organism is being fertilized by the sperm of the other organism. So why this is happening is that although both the organs although both sperm and ova are basically present in the same organism since they are hermaphrodite, but their production timing is different. That is, either the sperm will develop, the ova will not develop at that time, so this sperm will go and fertilize the ova of other organism, or vice versa may happen that the ova might have developed, but the sperm is not being formed or produced till now, and therefore this ova is being fertilized by the sperm of the other sponge. So therefore, it is important to know that although they are hermaphrodites, but they are being fertilized by the sperm or the ova of the other organism, and therefore they undergo cross-fertilization. Therefore, they undergo the process of cross-fertilization, right? So this is really important to remember that they undergo cross-fertilization. So the sperm and ova are derived from the undifferentiated amoebocytes, which are called as archaeocytes, right? So this is another kind of cells. We learn about choanocytes, we learn about amoebocytes, we learn about archaeocytes. So these are different cells that are having the different function. So the sperm and ova are also known to be derived from the choanocytes, which later undergo gametogenesis to form sperm or ova. So the choanocytes may undergo gametogenesis leading to the formation of sperm or ova. So the sperm or ova may be derived from the undifferentiated amoebocytes, which are called as archaeocytes, or basically amoebocytes are being attached. We know that the amoebocytes are attached to the choanocytes, right? So in some literature, you may find that the sperm or ova are also known to be derived from choanocytes, which later undergo gametogenesis to form the sperm and ova. So this is how sperm and ova are being formed. The spermatogenesis is the formation of the sperm, the process of formation of sperm. So in spermatogenesis, sperm is being formed. So how sperm is formed? The sperm mother cell or spermatogonium is the enlarged archaeocytes. So we learn that archaeocytes, which are the undifferentiated amoebocytes, leads to the formation of sperm or ova. So this spermatogonium is then surrounded by one or more flattened cover cells to form the spermatocyst. These cover cells are derived from other amoebocytes, and now these spermatogonium undergo 2 or 3 maturation division to form the spermatocytes. And then these spermatocytes later give rise to spermatozoa. So did you see how is this happening? So basically this spermatogonia is undergoing division, leading to the formation of spermatocytes. These spermatocytes then later give rise to spermatozoa. A mature spermatozoon consists of a round nucleated head. So they have a round nucleated head and they have a tail. So this is the structure of a mature spermatozoon, and the lashing movement of this tail helps the spermatozoa to reach the other sponges. So this is how the sperms we know are motile, and therefore they can, since they have a tail-like structure, and this is how these this flagella or the tail-like structure, not flagella exactly, but the tail-like structure helps in the movement of this sperm towards the other sponges. How oogenesis? Oogenesis is the process of formation of ova, right? So the egg mother cell or an oocyte is derived from large archaeocytes. We know even in literature also, whenever you will read about the ova formation or the sperm formation, you will always find that the egg cells are usually the larger cells. So they are derived from the larger archaeocytes with distinct nuclei. Sometimes the oocytes also arise from the choanocytes. These oocytes move like amoebocytes, engulfing the other cells. And the engulfed cells act as the nursing cells for the oocytes. So nursing cells means they provide food and nutrition to the oocytes. When they are fully grown, the oocyte undergo 2 maturational division to form the ova. Basically, the oocyte is undergoing division to form the ovum, right? Which lies in the wall of the radial canal or spongocoel, ready to be fertilized by the sperm of another sponge. So this is being present in the wall of the radial canal. You can see if you remember, suppose this is the spongocoel, this is the wall of the radial canal or the spongocoel. The ovum is present here, and this is ready to be fertilized by the sperm that is coming from the another sponge. The sperm from the another sponge fertilizes the ova, and the process is known as the cross fertilization, as I mentioned earlier also. So it undergoes cross fertilization. Now, how fertilization happens? Sperms are then released out from the sponge through the outgoing water from osculum. We know everything comes out with the water, along with the water through the cavity, which is the osculum, right, which is present at the free end of the sponge. The sperm that's released make their way into another sponge through incoming water by ostia. So whenever the water enters through the pores, that is through the ostia, along with the water, sperm enters into the another sponge. Choanocytes act as the nurse cells and transport the sperm to the ova, which lies in the flagellated choanoderms. The fertilization is internal. This is important. Fertilization takes place within the sponge, but within the body of sponge, but it is cross type. That is, it is a cross fertilization, and the fertilization is internal. That is, the fertilization happens inside the body of the sponge. This is a figure. You can see there. Here, the asexual reproduction budding is being shown. Here, the egg cells are being formed, and these egg cells, these sperm cells engulfed by the choanocytes. These sperm cells Here we have shown the sexual reproduction, where the fertilization between the egg cells and the that is the ovum and the sperm takes place. This fertilization leads to the formation of embryo. The embryo can be a planktonic embryo, that is the amphiblastular larva. Embryo develops basically into the larva. So this is the developmental stage where it develops into the larva. The mental stage where it develops into the amphiblastular larva. The larva settles down and attaches to the bottom of the other surface, leading to the formation of a new sponge. So this is how the sexual reproduction happens. Now these are certain examples of porifera. Example, cycon. You need to remember their common names also. This may be asked. So cycon, which is the scypha. Spongilla, which is a freshwater sponge. Euspongia, which is a bath sponge. Euplectella, which is a Venus flower basket, and Leucosolenia, which is the smallest sponge. Also, we remember that cycon and Leucosolenia are the ones that have a symmetry, which is the radial symmetry, right? So, cycon and Leucosolenia are the examples of poriferans. Spongilla, Euspongia, and Euplectella. You need to remember their common names as well. And with this, we end up with the phylum Porifera, and in the next class, we will take up the phylum Coelenterata or Cnidarians. So thank you, and see you all in the next class.